Very good. Well, welcome to the CSRC Colloquium. Today we have Jim Thorson from NOAA. So he is a senior scientist at NOAA Fisheries, and he's a program leader at the Alaska Fishery Science Center. Jim received a PhD from University of Washington in 2011. Jim has some skill expertise which is very relevant to CSRC. So he uses statistics tools for fishery and ecological research, and more importantly, is looking for collaboration opportunities at San Diego. And he comes to San Diego once in a while because San Diego has the Southwest Fishery Center at La Jolla. So, so if you're interested in this collaboration opportunity, possibilities for grant, supervising students, and of course writing papers, please contact me or Jim directly. Jim, you perhaps can type your email address right on chat, please. So Jim, welcome. It's your turn.
Thank you. Before I get, I'll share my screen, and can you confirm that you can see my PowerPoint?
Yes. Yes.
Okay, great. That NOAA has still banned us from Zoom, and so for some reason the interface was frozen a second ago. But please just verbally tell me if you can't see or hear me. Yeah. Without further ado, so thanks for having me. As Sam said, I work closely with colleagues at the Southwest Fisheries Science Center and the Inter-American Tropical Tuna Commission that's co-located at the La Jolla lab there. And 2 years ago, I took over leadership of this program that, among other things, tries to vision and coordinate ecosystem linkages to our fisheries work. The Alaska Fisheries Science Center is one of 6 of these NMFS labs nationwide. The Alaska Center has about 320 full-time employees and about 500 contractors, and maintains, by one count, 17 major field-going sampling programs on a biannual basis. And so we cover a tremendous area, the large marine ecosystems of Alaska and areas that are rapidly impacted by climate. So I'm interested in expanding our collaborations with climate scientists. The premise of this, I suppose, is that I see NMFS and NOAA and scientists worldwide putting a lot of effort, very justifiably, into Earth systems models and downscaled versions, regional ocean models for ocean dynamics. These are numerical models that have horizontal and vertical discrimination, run on weekly time intervals, solving physical differential equations, and parameterized to deal with small scale dynamics. And they have very good success in terms of forecasting physical changes up through primary and even sometimes secondary production. But when you move from secondary producers like passively advecting krill and copepods up to mobile animals are fitness optimizing. They are smart and have memory, and there's a lot of reasons why they cannot be easily modeled numerically in a deterministic model. And so I'm interested in building up sort of a set of spatially downscaled multi-species and ecosystem models that can be interfaced with regional ocean models or Earth systems models, but using the principles that we know work well for population community ecology, not just treating fish as sort of a little physical particle. Let's see. So in this talk, I will try to introduce what I've been calling the Grand Habitat Challenge. And then I have a series of different sections on a generalization of empirical orthogonal function analysis, or EOF analysis. The first showing this sort of generalization to kind of equivalent to a generalized linear model. The next being EOF for demographic processes, stomach contents and size and growth. The next being dealing with spatial transitions to interact with time series models. and finally doing a whole of ecosystem forecast using EOF. So again, we have an amazing track record in the United States of having ended overfishing, and we are in the process of rebuilding many fish stocks after decades of industrial fishing. Sustainability is typically defined for a population in a given area, which we call a stock. It cannot be defined at individual sites, and analysis of sustainability typically requires some environmental response, some understanding of how things respond to the environment, some assumed response to harvest or disturbance, and some understanding of density dependence, the ability for populations to have regulation where they increase in per capita productivity as populations become less abundant. And this definition of sustainability is sort of the basis of a lot of renewable resource economics and policy. It's written into policy at the UN and federally. And so a lot of these models end up being time series models in which you have a change in biomass B that is predicted from current biomass B. So delta B predicted from B given some density dependent function, some exogenous environmental response X, and some assumed driver catch C that's included as like an offset, an assumed exogenous driver of known magnitude. So for instance, you end up with these, you fit these models to data and you can get an estimate of biomass relative to some target and fishing mortality relative to some target. And that defines this sort of green space, the safe operating mode at which fishing mortality is low enough to be sustainable and the biomass is above some target. And we typically fit state space models to do that while trying to estimate these reference points, we call them. And so, yeah, again, this is sort of an explanation of how this works in practice. By contrast, in spatial modeling, we often try to identify habitats that are critical, and we do that using these sort of glorified regression models that we call species distribution models, or SDMs. And typically that just predicts a biomass, not a change in biomass, as a function of local environmental conditions at every site S. Where I can see I need to fix the subscripting of in the top row, but it's a different class of models, and I guess me and others have been generalizing to try to combine these two different traditions of models. So essentially building sort of a small scale biological model at each fine scale location S. And modeling changes in biomass, like in a state space model, given current biomass and density dependence, given some exogenous environmental drivers, and given some localized catches.
Motherfucking Christ.
And somebody might want to mute, and I don't know if there's a moderator who's able to mute other people who might have forgotten their microphone. Can anybody still hear me?
Yeah, you're good.
Yeah. Okay, great. So so future extensions, this is what I'm calling a grand habitat challenge to figure out how to do sustainability assessments with density dependence and environmental drivers and catches as a spatially downscaled model. To do this, we need extensions including state structure. It's okay. Yeah, no worries. So, future extensions could include stage structured effects, non-local effects like individual movement or environmental teleconnections, and mechanisms linking independent variables. So, for instance, we have, there's sort of mechanisms that associate temperature with depth and light levels with depth, and human changes, human actions might decorrelate the historical correlations that we've observed, and so we end up having to predict in a novel, what we call no analog systems, and so we need to disentangle the mechanisms among these independent variables as a causal statistics challenge. So just digging into the non-local effects, and this is what I'll focus on in the remainder. taking that sort of model I kind of quickly showed and trying to include sort of a saturated model in which every site affects dynamics at every other site, you quickly end up with this kind of graphical model that is clearly not identifiable because it's oversaturated. That, no matter how much data you have, as you do downscaling, you'll always get more more pairwise interactions that you might want to estimate, and there is no way to ever collect enough data to overcome that sort of infill asymptotics. So that the proposal, the kind of pragmatic proposal we've come up with is to deal with this sort of density dependence piece using kind of known ecological mechanisms for movement, so empirical movement models or an effective diffusive movement model based on local environmental conditions, and then trying to do feature identification, so ways of doing rank reduction on covariates to come up with principal modes of variability and how to include those. And so feature identification, we use PCA or EOF, and the remainder of this will be really talking about how EOF is sort of a way of doing spatial feature identification to deal with spatial variation and non-local effects. So in the first paper here, this is a paper from last year with Lorenzo Cianelli at OSU and Mike Lutzow, who's now at the Alaska Fisheries Science Center. And I like to set it up by thinking about this Lake Alignagik in Alaska. This is facing north, and essentially, if you put out stream gauges to sample temperature in the headwaters or the outflow, a kind of simple box, like differential equation box model, you might think that all of the headwaters will have highly variable temperature due to melting conditions in the headwaters, and the timing of melt, cold water from melt. And that they'd all mix and average, and the outflow would be more stable than the inflow and would provide this thermal refuge. In fact, if you put out gauges, the outflow is stable up until the early summer, and then it suddenly gets kicked and starts oscillating at this multi-day frequency from cold to warm. And after thinking about it, people realize this is satiating. This is where in the summer, as the surface heats, it sets up what's called a thermocline. And then when the first storm passes through, it pushes all of the warm surface water to one end of the lake. When the storm abates or passes, it sets up a rocking, like the rocking of a bathtub, where when the thermocline is above the outflow, it dumps cold water. When the thermocline is below, it's warm water, warm surface water. And so it's an example of these sort of physical characteristics setting up characteristic modes of variability and affecting the thermal environment that species have to adapt to climate change. So we started out wondering, how do we identify annual variability from multivariate, noisy, zero inflated biological data? And we're going to proceed by doing this additive decomposition of spatio-temporal data into one or more time series and a spatial response for every species. And this again is just a generalization of EOF. So if you look at something like the Pacific Decadal Oscillation and the El Nino Southern Oscillations defined similarly, it's a way of decomposing surface temperatures into a mode of variability in the bottom, and then a response map in the top, where, for instance, a warm phase, it shows higher than average temperatures off the coast of Alaska and Oregon, California. The cool phase is the inverse of that map. And at the time I did this screen grab in 2017, we were in a warm phase. So it's a way of taking this sort of spatio-temporal data and crunching it down to a single feature composed of an index and a map. For sake of time, I'll skip that. So, one way this is generalized is by replacing replacing the map with a Gaussian Markov random field. So in this case, we'll have omega is my convention for a spatial term. Omega is indexed across space S for each factor F, and you can sum across a number of factors. where every response, and if you have multivariate data, you can have a loadings matrix that associates each response variable with each factor. So L is a loadings matrix, similar to how conventional factor analysis works. And treating it as a Gaussian Markov random field means that at a finite set of points, it's multivariate normal. And there's well-known ways of approximating, a Matern correlation function as a Gaussian Markov random field as a sparse precision matrix. And so that's why I proceed that way. And then further, this can be generalized by, applying a link function and having an exponential family distribution for data. So this ends up being a sort of like the equivalent of taking a linear model and making a general linear model. It allows you to model count data or a variety of responses, and using a log link or a logit link. In the remainder, we'll end up doing what's called approximating a Tweedy distribution. But just to continue this example, I want to show that we know what we're talking about. I've skipped the North Pacific gyre oscillation, which is calculated from sea level height, not temperature, and so it's not perfectly correlated. But in terms of the first mode of variability, the PDO, just taking an official value from online at the time, and instead running an estimate on a net CDF from online of surface temperature, I get a very high correlation, 0.92, and the estimated map in the bottom left, to my eye looks a lot like the warm phase that's shown on that kind of downloaded image from online. So I hope that I've convinced people that this is sort of a valid generalization of EOF. So next, skipping the rest of that paper, I'll just skip to more recent work that's in revision at Progress in Oceanography, working with Arno Groos, who's now at NIWA in New Zealand, and then Christine Stawitz, John Ream, Sean Rohan, and Cheryl Barnes are all in one way or another associated with the National Marine Fisheries Service. And in this effort, we're trying to synthesize a bunch of different information about spatial demographic processes to see how regional ocean conditions affect a $1 billion annual industry, fishing, Bering Sea, walleye, pollock. We catch about 1 million tons a year of it, and anytime you've ever had a fish stick, that'll be Alaska pollock. It's about a 1% of global fish production. So, for instance, we can take what a relationship that we often analyze in fisheries between weight and length. So, body weight W is often assumed to be this allometric, this power function of individual length L, with an intercept beta, and this is sometimes called Lecrane's condition factor. Condition is essentially measuring how skinny or fat a fish is, and as they get fatter, they have energy to put into reproduction. The fishery also wants to catch them because they have more muscle mass. And so there's an economic and an ecological reason to care about condition as an integrated measure of environmental success and fitness. So if you take log of both sides and then add a bunch of spatial and spatio-temporal elements, you get this lower component, this lower decomposition, and I'm not going to walk through it in detail, but it has a spatial correlation omega. It has epsilon, which is my notation for spatio-temporal variation, a spatial component that varies among times. It has a loadings matrix for across time, which is this EOF decomposition. And then it also allows us to put in covariates, either with a linear effect or a spatially varying slope, like a varying slope model in kind of Gelman terminology. And so we can look at sort of how much observed patterns are explainable as a function of covariates. So we're going to do this for morphometric condition. I said that. For growth, the average size at age. For consumption, or prey biomass in the stomach samples of a given of pollock as a predator, and also numerical abundance to represent movement of pollock within the Bering Sea. So each of these is sort of a basic data type that represents a different biological process. And all of them can be treated as a univariate or a multivariate generalization of EOF. And specifically, we're going to explore exploratory factor analysis, sort of this EOF model, but also what I'm calling confirmatory factor analysis, where we put in a known covariate, and it enters very similarly to the loadings matrix. And we can see how much we can explain a mode of variability that would come out sort of descriptively as being due to a known annual covariate. So I'll skip that for sake of time. So each of these processes, we can get a spatial map representing the distribution, spatial heterogeneity and density, condition, length, or stomach contents in an average year, a year where the loadings matrix is 0 for every factor. And this is a snapshot of Alaska. So there's like this is the Bering Sea, there's Bristol Bay. You can see here, this is called the Aleutian Peninsula, and this is the North Pacific. Russia's up here. And numerical density in an average year has highest density in what's called the this kind of northern outer domain. Fish are fattest in the south. The length for a given age is highest out here in terms of in unit centimeters. And then they consume the most copepods, the little crustaceans on the outer domain, the most euphausids or krill in the south, and the most Mysidaceae or shrimp kind of into Bristol Bay. So you can start seeing what they're eating in every area of the Bering Sea in an average year. More interestingly, you can also get these modes of variability that are estimated. So in these cases, this is showing modes of variability. In the top row for the numerical density. And for numerical density, the major mode, one of the major modes of variability has a 0.75 correlation with what we call cold pool extent. So cold pool extent, there's an island called St. Matthew's Island. It's an ice factory or a polynya where predominant wind pushes, it's cold, sea ice is formed. When sea ice is formed, it causes salty, rejects salt. And that salty cold brine sinks to the bottom because it's dense, full of salt. It's a polynya where air, wind is pushing that ice away from the land, and there's nothing to move into there because there's land. And so it continues to produce ice. And because it's doing that, it makes all this cold water that spills south into the Bering Sea. The spatial extent of that cold pool that's formed every winter is sort of a metric of cold water that we show here when the cold pool is large, there's lower density where in the north where it's being formed, and they redistribute offshore a little bit. On the right hand side, this is sort of if we take the cold pool extent and put it into the same model, what's the response map that we get? This is sort of maps of the weight at length or condition in response to cold pool. So they have lower condition when the cold pool is large, and they have higher condition in the south. This is the response map of size. So in a year with a low cold pool, they have, I guess when it's a high cold pool, they have higher size near shore, and vice versa when there's a large small cold pool. And then in terms of prey, when there's a low cold pool or a positive response, I guess they eat more copepods kind of near the Pribilof Islands, more krill in the Bristol Bay, and more shrimp kind of in mid-domain. So you can start seeing these sort of ecosystem impacts from sort of this confirmatory factor analysis model. So so the conclusions of this, we can do sort of these essentially sort of spatially downscaled models of ecological processes. And we can start trying to ask questions about how regional environmental conditions, like in this case, the ice factory producing a cold pool, how that affects habitat utilization for this fishery, this stock that supports $1 billion industry. It also confirms that there's this strong bottom-up oceanographic driver for Alaska pollock. And more on that in a second. So I've got two more kind of examples, and I'm kind of going through these quickly to make sort of a broader point. This one is published last year in Global Change Biology and co-authoring with Wei Cheng and Al Herman, who were regional ocean modelers in Seattle. Jimmy Anelli, Mike Litzo, Cecilia O'Leary, and Grant Thompson work with me at the Alaska Center. And we're interested, this is a picture of ice being infected south from the Bering Strait. And then of course these are larval fish. And so larval fish operate on these sort of, they starve or they get eaten on sort of a time, characteristic time scale of minutes to hours. And, they can only move over, meters to hundreds of meters. Actively, whereas we're talking about regional processes occurring across, at a basin scale, at a global scale. And so there's this interesting sort of scale transition you need to do to link regional survival of larvae to regional oceanographic processes. So to do this, this actually, maybe I'll skip this, but this is sort of a slide about EOF again, just kind of summarizing what we said. There's this spatio-temporal process epsilon that's decomposed into a loadings matrix L and a response map for each factor. And we can do EOF, a generalization of EOF with that. Where this gets interesting is if we take that, the next step in what we study in this paper is if we take, the first mode of variability and then also put it in as a covariate in a time series model. In this case, just a conventional linear model, and define a distribution for the measurements. And that's a way of coupling EOF to kind of a basic kind of time series and linear modeling. This is helpful in fisheries specifically because one of our ways of modeling the survival of recruits, this is what, survival of larvae and eggs, is called the Ricker stock recruit model. And it's a linear model. The log of recruits per spawner is a linear model of spawning biomass. So in this case, we can take a well-known Ricker stock recruit model and couple it with an EOF of regional ocean conditions, and that's what we do. So as kind of a pictogram, again, we've got these sort of one or more physical components, physical variables that we have measured. We decompose those into a time series, the EOF index and these response maps, but then that EOF index is also shared in a linear model for some time series biological process. And we're going to fit this to bottom temperature measurements from over 30 years, 35 years, surface temperature from satellites in the winter, and then Earth systems model forecasts of those to end of century climate conditions. And we're also going to fit that with biological data, so stock and recruitment records for three species, pollock, like we looked at before, Pacific cod, yellowfin sole. These are three of the most commercially valuable stocks in the United States. So if we do this, as one waypoint that I won't talk too much about, we're essentially doing what's called a delta change method or a delta correction method to correct for biases in the regional ocean modeling system that we're using to forecast. So there's a period of overlap where we have measurements and we have a hindcast from our Earth systems model, and we can look at how those vary and then basically deal with the average difference at every location between the measurements and this Earth systems model, and allow that to do this sort of delta change method and correct for these. And anyway, so if we look at bottom temperature or sea surface temperature, either the predicted observation or the predicted model output, the observation, the model output patterns look quite similar in general, like the first column and the third column, for instance. And by 2090, in the bottom row, we have this tremendous warming of the Bering Sea, particularly near shore in Bristol Bay, and that's no surprise. If we then take that model and we couple it to stock and recruitment records and visualize the map that we get, that's essentially the mode of variability in the physics that's maximally correlated with deviations, positive survival. And so in this case, pollock and cod have sort of similar, I'm just going to look at the bottom temperature response maps. These are the maps where if physical conditions look like this, you're expecting to have higher recruitment. And so pollock and cod both have higher recruitment on average when there's sort of this cold water in the southern middle domain, this kind of blue area. And that occurs when there's a large cold pool extent. We know that cold years are associated with good survival of pollock and cod. Yellowfin sole has essentially the opposite, where that same area, if it's warmer than average, has good recruitment for good survival of eggs and larvae for yellowfin sole. Pollock and cod don't have the same picture. Cod has kind of cued, it has more influence of nearshore cold waters, and that makes sense given that cod have sort of spawning grounds up in that nearshore area. And then, we can use this model to forecast all of these stocks out into end of century conditions, either with this EOF linkage turned on or leaving it off, and assuming that current spawning biomass stays the same for the purposes of this forecast. And for cod, for instance, this top row, the correlation using the EOF is 0.68 compared with a correlation of 0.60 without using environmental conditions. And then it forecasts in log space a reduction by about 1 of recruits per spawner. So there's about a 30 to 40% as great a reduction of 60 to 70% in their survival of eggs and larvae by end of century climate conditions. Pollock also has a reduction, and yellowfin is expected to have positive responses to warming, assuming that everything else stays similar. So EOF, what I'm calling EOF regression, is a way of generating hypotheses about multi-decadal changes and identifying environmental conditions that are associated with a given time series. They're also useful, I think, to visualize and communicate time, do this linkage of spatially integrated biology to spatially downscaled physics, and could be used to localize, prioritize localized monitoring programs. So like, I gather that there's a, San Diego State has a, carbon dioxide monitoring at Utqiagvik, formerly Barrow. Cases like that, we spend a lot of money in locating monitoring efforts, but those efforts aren't necessarily I don't know what the justification of locating in Utqiagvik is except logistics. And so there's sort of a benefit to formally identifying what's the best place to monitor a given regional mode of variability. And then finally, just to kind of continue quickly through. So this is the last one. This is sort of an example of using EOF for whole of ecosystem forecasting. And this is with many of these same people again, but calling out Aramitsu, Yumi Aramitsu, who's at USGS, a seabird expert, Louis Barnett, Lisa Eisner, Alan Haney, Kirsten Holzman, who does a lot of climate work, Yvette Sidden, John Richard, Mike Lomas is out east, and Dave Kimmel. And so this is a huge effort to assemble a bunch of ecosystem data in the Bering Sea and think about how to link it to climate. And it's online, but not yet paginated at Ecological. So in this, just continuing on, we assembled some of these data we've shared before. So summer bottom temperature, winter surface temperature from satellites, Earth systems model forecast of those. We saw that in the EOF regression example. Here we're also adding primary producers, both small and large, in the spring and the fall. Secondary producers, Calanus, Glacialis, and Marshallet. That's this copepod, this small crustacean that is a key pathway. These crustaceans eat little phytoplankton, and then they're big enough that they're valuable to eat for fish. Juvenile pollock that are up near the surface, you can caught with these surface trawls. There's the bottom trawl of pollock cod, snow crab, tanner crab, and arrowtooth flounder. There's 2 seabirds, shearwater and northern fulmar, that are witnessed in these kind of essentially citizen science efforts out on shipping vessels, vessels of opportunity, and then catcher-processor fishing effort. These are these big industry vessels that go out and harvest pollock, and we have a record of where they're fishing. And if we look at data availability, we end up with these thousands of samples per year. This is I mean, given that a single bottom trawl sample might average and cost, $1,000 to $10,000, having hundreds of thousands of samples is a tremendous investment of money that goes into it. The core of our sampling is in the summer, but we also have fall and spring data. And looking at the component that we have the most data about adults and seabirds, adult fish and seabirds. But we have all these ecosystem components, primary and secondary producers, and juvenile fish, kind of starting in the mid-2000s. So looking at these kind of conceptually in terms of vertical availability and season, we can kind of see that we've got this pulse of spring and fall data that's ecosystem oriented, and then the summer is our kind of core adult bottom trawl data. Yeah. So, okay, I forgot to add some equations. I'll skip that. But let's see. So this is the map of average spatial conditions for each variable in the model. So again, bottom temperature is our coldest in the northern area. Sea surface temperatures look similar to what we've seen before. The bottom temperature in a regional ocean modeling system and sea surface temperature, that's doing the delta change method to sort of line up our measurements in our ocean model. In terms of chlorophyll, we see in the spring, the second row, second and fourth from the left, we see this high density of chlorophyll in this area that's right off Jemcha Canyon. This is the beginning of what's called the Bering Sea Greenbelt, where deep waters are advected up, they're nutrient rich, and they fuel this bloom of phytoplankton. Secondary producers in the spring and the fall, and then we've got these different fishes. I won't get into it too much, but if we go to the bottom row, the fall catcher-processor fleet has a distribution that looks a lot like the distribution of pollock in the third row, which is what they're catching. And it makes sense that in the fall, they're lined up with the summer distribution of what they're fishing. So let's see, sorry, yeah. So the dominant mode of variability for this has a 0.8 correlation with the cold pool extent. So, but also because we've got these forecasts, we're doing a decadal scale forecast of this mode of variability for which we don't have a measurement of cold pool extent. And our Earth systems models kind of, are projecting that warm conditions will continue, albeit with a great deal of interannual variability and uncertainty among models. So the map of responses to warm conditions look like this. So in the third row, pollock in warm conditions move north, so does Pacific cod, so does snow crab, so does arrowtooth. In warm conditions, the spring chlorophyll has lower density in Jemcha Canyon. The greenbelt is less strong, seemingly. And I probably won't get into too much of these other. Well, and that I'll say the catcher-processor fleet in spring, when there's still sea ice. Spring is when they have sea ice, but in warm years there's less sea ice, and so this fishery is able to go further to the north without risk of harm to their vessel. So we can also run this model into the future, and it's essentially like a forecast of the distribution of every species. When we do that, we get this sort of picture that fall Calanus will have lower density over time. That this eggs year pollock, these sort of young of the year pollock, will be distributed across the Bering Sea, and that arrowtooth, this sort of voracious predator, will continue to spill from offshore inshore. So there's essentially this invasion of a new flatfish into the Bering Sea. And then finally, there's sort of this metric of overlap that ecologists use called Schoner's D. We can calculate overlap for every year, and if we do this, we see that, well, essentially there was sort of this early 2000s. There was a warm, what we call a warm stanza, and then it switched to a cold stanza from say 2005 to 2012. I guess I have years listed here. 2007 to 2014 is what I said here. Cold, and then it went back to warm. It warmed rapidly from 2015 through 2019 and 20. And so in these in the two warm stanzas, there's less overlap between Aegzir pollock and its major prey of fall calanus. When it's warm, there's more overlap between adult and Aegzir pollock, which is an issue because they're cannibalistic. When it's warm, there's more overlap between adult pollock and its major competitor, arrowtooth. And then when it's warm, there's some changes in the overlap between, shearwater, a seabird, and the fall catcher prosser fleet. So in conclusion, like anybody on the West Coast, I spent a lot of time thinking about forest fires recently, and that's a perfect example of these ecological teleconnections where atmospheric emissions globally are creating a situation that has extremely localized impacts. There's fires and drought are primed by basin scale oceanographic features like ENSO cycles, and then of course, like living in Seattle, I've got a 7-month-old, and we had this sort of plume of smoke right around the time when she was born, and so this is also a teleconnection where there's a mechanism linking sort of air quality in Seattle to tragic fire conditions that are more immediate for you guys. So these sort of teleconnections are well known in oceanography and atmospheric science. They're less studied in ecology, I would say. And they're one way of addressing non-local effects, the other being these sort of explicit individual movement models. And then I guess I've got a separate set of talks about these sort of causal statistics and independent, how to deal with changing association among, independent variables as well as stage structured effects. I'll say that we are often looking for postdocs. I mean, these, I'm kind of narrowing in on filling, but I think we still have money for 2 years of the postdoc looking at climate impacts on zooplankton, and feel free to reach out if you're interested in statistical ecology in particular. And again, I'll put up some, I forgot to update the top one, but I should say in particular, thank you to Sam for inviting me. And thanks everyone for listening.
Good, thank you. Good job. I really enjoyed it. I listened to you second time. Every time I learn something. So questions for Jim. So for your EOF prediction that's using the principal component, have you looked into a method actually invented by SIO researcher Tim Ballett? It's called a canonical correlation analysis. Well, that's a statistics method, right? But he introduced the EOF into CCA and then really changed the way of climate forecasting and climate prediction at CPC, NOAA CPC. But have you tested that method?
No, I haven't. And I should say that, I think there's I was looking at CCA. I was also looking at canonical correspondence analysis recently, and I'm even forgetting what the distinction is between the two right now. There's a lot of there's a whole toolkit of how these methods are used in community ecology, like taking a vector of species samples and using Bray-Curtis dissimilarity to convert it to a distance matrix and then running it through methods like you're saying. And I'd love help thinking about the kind of community ecology interpretation, and I'd also love to catch up on how physical climate modelers are using the same things. I would love that. Yeah,
That'd be good because you know, CCA, that's the maximize the pattern correlation. You know, they normally we just say two time series computer correlation, and then you have two patterns involved, and then you compute correlation, and what will be the best pattern. So that will be very much related to what you're doing, and it might be interesting. That's another thing for this audience. I just wondering whether you want to explain a bit more about EOF from the point of view of space-time decomposition, because that is something we have a lot of people who are interested in mathematics here. That you know, how to understand data from the space-time decomposition point of view. You said it really well. Do you want to say a bit more?
Sure. Yeah. I mean, so I'll say that, I mean, you know, and I'm not an expert, and I think you, Sam, probably are that expert. And so feel free to jump in if you disagree with any of this. But, you know, my experience with EOF is that, let's see, I guess I should share my screen if I'm going to use it as an example. And everybody's frozen again, so I can't see anybody on the camera, and I'm sorry that I can't take visual cues well. So yeah, so I'm used to people taking, you know, downloading these netcdf files that have sort of, you know, a value at every space, you know, in a sequence of years, and you can, you know, my experience is that people typically subtract off the mean for a given site. You know, well, first of all, that's spatially balanced data, and it's convenient, you know, in the satellite age because we've got, you know, high quality measurements and people can Krieg them and make data products online. You know, if you don't have spatially balanced data, it's harder to apply these sort of out of the box. And that's one benefit of generalizing it to have a Gaussian Markov random field. You know, setting that aside, you know, people take these spatially balanced, you know, data sets that they get online and subtract the mean from every raster cell. And by doing that, you know, as an ecologist, you lose something useful, because in ecology, there's often a mean variance relationship, you know, in what's called the nugget, like the geostatistical nugget. And so you don't want to just modify the mean without also thinking about the variance in ecological data. You know, so that's another way that sort of classic EOF doesn't generalize well to community sampling for ecology. But if you do that, just subtract off the mean with spatially balanced data, you get this sort of matrix of deviations for a single variable at every space and time. That's a matrix, and you can decompose that matrix into, you know, the outer product of a time index and a spatial response.
Very good. Yeah, that's very good explanation. We have people doing the image analysis here, AI, etc. You know, we really pay a lot of attention to the algorithm of SVD, singular venue decomposition. That's exactly this. So you mentioned about letter CDF. We also look at where people are interested in big data. You know, the letter CDF is really a popular way today in writing space-time data. You have space every point, and then time. And singular venue decomposition, you know, come out with U matrix, that's space, and V matrix as time. So that's what you should be aware of. The U matrix will be your global picture, and the V matrix is your the time series. That's really nice. Yeah. So,
yeah, and you know, like I just want to stress. I mean, like I think, you know, there's sort of a lot of like computational optimization that goes into optimal ways of calculating SVD. You know, and if you have spatially balanced data, you know, that's presumably the most efficient computational way to do it.
Yeah.
You know, you can generalize all this as a hierarchical model, statistical model, and you know, modern computation uses these sort of sparse matrices, and in a lot of times you can actually get really close to efficiency, you know, theoretical efficiency using this statistical generalization. you know, with sparse matrix like the eigen package in C++.
Sure. Yes. Great.
So there's some kind of interesting kind of deep math about how to, you know, how to do that generalization efficiently.
Thank you. Jose, you have a question?
I don't have any questions. I think that was very nice.
Okay.
So, but
I have a question.
Hi, Chris.
Hi. Yeah, so correct me if I'm wrong, but I believe that in EOF analysis, the idea is that each empirical function kind of represents a distinct spatial process, like an underlying process in the data. If that's the case, then it seemed like throughout your talk that you were really focusing on just like the primary or like the main mode, the first EOF. Is that just because it's like way larger than the other ones? The other ones are insignificant, or like does it get complicated when you introduce more than one?
Yeah, I mean, honestly, like I'm still trying to convince. So that's a great question. You know, like I'm still trying to convince my colleagues that we should spend more time thinking about EOF for our biological sampling. And again, like we haven't typically done it because of these sort of mean variance things and this spatially balanced stuff. You know, and so it's helpful rhetorically for me as an ecologist to point out that there's sort of kind of, you know, like for instance, cold pool, we spent $40 million in a Berserk program with NSF to identify that cold pool extent was the dominant mechanistic driver. And so it's nice for me to take publicly available data and just kind of show that as a mode of variability. You know, so I'm trying to build confidence in the first mode before trying to interpret the other ones. But you know, like we do see these sort of second and third modes that are real head scratchers sometimes, or other times we're in the room with somebody and they're like, oh, well, that kind of looks like this. And, you know, it's not, you know, then you just set up more experimental and process work to figure out if we trust what the other ones look like.
Very good. Yeah. Any other questions?
Thank you.
Yeah, thank you.
Okay, so look like we are done. Thank you very much, Jim. That was really good talk. And then thank you for the opportunities of a postdoc and also your offer to collaborate. So make sure that you visit us when you come over here next time.
I will.
Pandemic.
Yeah.
and could how do you call it? What kind of skills are you looking at? What kind of postdoc?
Yeah, I mean, I call myself a statistical ecologist, and but you know, we do work a lot with like, you know, like Earth systems modelers and computational biologists and people who, you know, we typically use R, but we use C++, and we try to use, you know, think kind of about hierarchical modeling, but also about sort of simulations to deal with big data. I don't know. You know, I'd be curious to kind of hear more from anybody who's interested in reaching out.
Okay.
Good.
Well, our students, let me just to describe the profile for students in general. Our students in general are able to identify, working a problem coming from sciences and engineering, formulate mathematical models, develop algorithms, and implement those in hopefully high-performance computing environments. They are tool makers as much as tool users, because most of the packages that are available, they don't solve the more important problems. So, and they are very, how do you call it, flexible. They can work as an individual, but also as a team leaders or team members. And they have a solid background in computational and data science skills. So,
Sure.
Yeah, and also, you know, we have our students are very skillful in high performance computing. We have a supercomputing class that is very helpful, you know, when you get to the huge data metrics, and also you may get into, you know, later stage, your team may be interested in machine learning. So that will use our supercomputing skills. Some of our students can do that.
Sure. Yeah, I mean, I also work with kind of big data for life history theory and kind of evolutionary theory, and there's, you know, then we have kind of similar things with kind of genetic, you know, huge data for genetics, and there's a bunch of cases like this, acoustics too, generating terabytes and you know, petabytes of data. So, you know, anyway, I mean, like I said, if people are interested, I'm kind of curious to just kind of follow up by email.
Great.
Okay, so I'll point it to your direction, and then you will talk to them.
Well, yeah, I mean, I'm always happy to chat
Well,
by email.
I'm just wondering if you have like a description. So I'm looking to work.
Well,
with
yeah,
this.
right. I mean, again, like so I do have descriptions for those postdocs, but I also do visioning for the whole Alaska Science Center, and it's, you know, at any given time we might have 20 postdocs advertised. So, you know, I don't know what to say exactly, except that, you know, I suppose I could send a description for that one postdoc that I'm currently advertising, and then I kind of, you know, just invite people to follow up if they're interested.
And this is to work in Alaska or in Washington?
We typically, yeah, I mean, typically I work, you know, people are located in Seattle or Alaska, but we have a lab in Oregon, and then, you know, these days it doesn't exactly matter where people are.
Yeah, but that's not gonna be forever, you know.
Yeah, well, exactly. I mean, you know, and then I work with people, you know, in other places, but yeah, typically if I'm funding them, they're people that live in Oregon, Washington, or Alaska.
Okay.
Actually, Jim is looking for people in the sense that, you know, if you're interested in doing this type of research, you just email to him and then start a discussion. And Jim then can organize a project for you, opportunity for you. So that is another option, and that option may be even more productive.
Okay.
Yeah, well, and I'll send that one description to you,
Okay.
Sam, and you can.
Yeah, that would be nice.
Sure. Great.
Okay, well, thank you very much, and have a wonderful weekend, everybody, and thanks for attending the talks.
Yeah, thank you, everyone.
Bye-bye. See you guys next week.
Bye-bye.